In one of the previous videos on my channel, we took a very global, very general view of the coca plant and saw how it manufactured cocaine. In this video, we're going to look very specifically at the biosynthetic pathway and see how the coca plant, specifically within the leaves, performs a biosynthetic transformation of lysine, a simple amino acid, into cocaine, which you can kind of see right here a little bit translucently. We'll get there at the end of the video. Now, as I implied, the biosynthesis of cocaine takes place within the individual leaves of the coca plant, and it all begins with this simple amino acid that we learned about in introductory biochem, which was lysine. Here's its chemical structure right here. Now initially there's an enzyme called lysine decarboxylase that removes the alpha carboxyl group from lysine. And that gives us this molecule called cadaverine. Now there's a similar molecule that you may have seen in other courses called putrescine, which actually gives the next enzyme its name. But this enzyme can act on both putrescine and cadaverine. In any case, putrescine N-methyl transferase uses the methyl donor S-adenosylmethionine, or SAM, to transfer a methyl group onto one of these amino groups. Okay? So that turns cadaverine into N-methyl cadaverine. Then we have this enzyme, N-methyl putrescine oxidase, which catalyzes the oxidation of this carbon right here. Notice that this is the carbon that has an amino group which lacks the methyl group. So it's just a simple amino group. You end up losing the amino group and it's replaced essentially with a carbonyl right here, more specifically an aldehyde. Now I don't have this molecule named because as soon as it's formed, very quickly and non-enzymatically, it's cyclized into this molecule called N-methyl pyrilinium. It's more stable for this structure right here to be a 5-membered ring, and so the lone pair that's on this nitrogen quickly attacks this carbon, and you end up with Schiff-base formation and elimination of this oxygen as water. And now you have the more stable N-methyl pyrilinium, and notice that this nitrogen right here has 4 bonds, so therefore it has a positive charge, and it can actually accept lone pairs, which it will actually do later on throughout this reaction scheme. Once the coca leaf has formed N-methyl pyrilinium, it's completed the first phase of the biosynthesis. There's really 2 more phases of the biosynthesis. The second one leads to the formation of the backbone of the cocaine molecule, which is essentially the molecule Echonone, which we have not seen yet, we will. And then the third phase takes that backbone and matures it into the molecule we know as cocaine. So the next steps involve this enzyme called Pyrilidine-Ketide Synthase, or PYKS, C-acetyltransferase. Kind of a mess of a name, and it forms a mess of a molecule. And so essentially what happens is this N-methyl pyrilinium, cation, is condensed with the acetyl group of acetyl-CoA. That's what this enzyme does. And it forms this molecule right here, 1-methyl, 2-pyrilidinyl, 3-acetyl-CoA. Now both of these molecules have this horrendous name up here. However, you'll notice one of them is the R stereoisomer at this carbon, the other is the S stereoisomer at that carbon. Both of them are actually going to be used in a subsequent reaction of this enzyme, Pyrilidine-Ketide Synthase C-acetyltransferase. Essentially what's going to happen is the acetyl group right here of the R isomer is going to be transferred to this carbon right here of the S isomer. Okay? So this carbon right here and this carbon, remember acetyls are 2 carbons, are going to be condensed here with this carbon. And you essentially form this acetyl-acetyl-CoA type of derivative right here, which has the name 1-methyl 2-pyrilidinyl, 3-oxobutanoyl-CoA. Oxobutanoyl is another name for acetyl-acetyl-CoA. This molecule is then going to be oxidized by a P450 type of enzyme. Essentially what's going to happen is this nitrogen-carbon single bond is going to be oxidized into a Schiff base right here, as you see. And then you get this carbon right here, which is deprotonated. The deprotonation doesn't occur by the P450. However, it's fairly easy to deprotonate this carbon and form a lone pair right here. And the reason for that, number 1, is that that lone pair would be stabilized in resonance with these 2 carbonyl groups. So intrinsically it's already simple to deprotonate there. But also, if we consider the actual conformation of this molecule, it actually looks like this. So notice when the P450 forms this Schiff base, this nitrogen has 4 bonds and therefore has a positive charge. And so if this lone pair on this carbon attacks this carbon right here, then the pi electrons in this double bond will simply go onto the nitrogen, and you will effectively cyclize the structure, making it more stable. So again, this carbon is incredibly easy to deprotonate because of the resonance stabilization, but also this nitrogen can act as an electron sink, which makes this carbon an incredible electrophile, especially when you have the molecule that's shown in this conformation. Okay? And that's exactly what happens. So it cyclizes into this molecule right here, which is called echanoyl-CoA. That completes the second phase of the synthesis. Now we have the echonone backbone, and we're simply going to do some transformations to it to form cocaine, which, by the way, has the technical name benzoyl methyl echonone. So once we have the cocaine base structure right here, we're going to have a hydrolysis reaction by a hydrolase that's unnamed, and it's going to remove this coenzyme A group. Coenzyme A floats away, and now you have a carboxylic acid right here. Now, this oxygen of the carboxylic acid is going to be methylated. It's going to be methylated by an O-methyltransferase reaction, as you would guess. The methyl donor is going to be S-adenosylmethionine. Now you have a methyl group there. This molecule is aptly named methyl echonone. Then we have an enzyme here called methyl echonone reductase, which uses NADPH to perform the reduction reaction, and essentially it targets this ketone right here and reduces it to an alcohol, or an OH group. This molecule is methyl echonone. We're almost done. We just have to now combine the methyl echonone with the benzoyl group right here, and the benzoyl donor is benzoyl-CoA. So then the final reaction here is catalyzed by the aptly named cocaine synthase, and it essentially takes the benzoyl group right here and adds it onto the oxygen here of the hydroxyl group in methyl echonone. Therefore, when you attach the benzoyl group on this oxygen of methyl echonone, you end up with the name benzoyl methyl echonone, which has the common name cocaine. And this entire biosynthetic process takes place within the coca leaves of the coca plant, which explains why, when cartels are manufacturing cocaine, they have to have an unimaginably high quantity of these leaves. And if you want to learn more about that process, there's lots of shows on Netflix that are very educational, and I'll try to remember to put a link to one or two of those in the description of this video. But hopefully here you learned a lot about the biosynthesis of cocaine on the molecular level. Thanks for tuning in. Please like, subscribe, and check out my Instagram for cool science and not science stuff.